Biology 107, Lab 5, Eukaryotes, Cell Organelles, and Cell Motility. In this week's lab, you will examine the actions and roles of various organelles and the cytoskeleton in three protists, Paramecium, Euglena, and Pelomyxa. The first activity will be to observe the swimming behavior of Paramecium caudatum and attempt to identify the structures responsible for its motility and other organelles such as the nucleus. Here are a couple of pictures that I took of some paramecium. This is under the 40 times objective. And those are some of the best pictures I was able to get. They're not too bad, and you can actually make out a few different features around the parameciums. So I'll point out a couple of things here, and maybe you can see some of these same images in the videos and other photos that I've provided. If you take a look at the paramecium on the left, I think what I'm looking at here is the nucleus, at least the macronucleus. You can maybe see some other organelles in there. I can see a smaller organelle here. I'm not entirely sure if that second one is the contractile vacuole or a food vacuole, but some sort of vacuole in general. The other thing that you can maybe make out, well, the one on the right as well, I think that might be the nucleus. It's a little hard to tell from that particular image. But one thing I wanted to point out are these structures on the outer edge of the paramecium. The one on the left, you can see the structures are kind of blurry. The one on the right, you can almost make them out. And those structures are constantly moving. And those are the cilia that are involved in the paramecium's motility. What you're going to do for the first activity, like I said, is to observe the swimming behavior of paramecium, and you can take a look at that in these three videos. There are probably other videos on YouTube that you can find. What I was trying to do is find videos that were somewhat similar to what you would observe in the Biology 107 lab. You can see on the left there's one swimming there. There's some questions in the lab manual to answer about that, and your goal is to identify the motility structure or structures. Additionally, we will observe the ingestion of food and the formation of food vacuoles in the paramecium. In the video, paramecium feed on yeast that have been stained with the vital dye Congo red. Congo red is a pH indicator dye that is red in color at neutral pH, and then at acidic pH it turns a yellowish orange color. As food vacuoles fuse with a lysosome, a change of pH occurs, and this color change will enable you to directly observe the process of digestion in paramecium. Take a look at those two videos there. The one on the first one is a little bit more like what we would observe in Biology 107. The second one is a lot more professionally done, and it's a really stunning video to look at. As a part of this exercise, I would also like you to measure the size of each of the specimens that we're looking at and draw a scientific diagram. Here's an image. It was a lot better than the images that I took, so I borrowed this one for this exercise. You can see that you can see both the ocular micrometer and the size of the field is shown on the actual diagram. So you can use either method to determine the size of your paramecium, and you're going to enter that in table 5-1. Also draw a scientific diagram, and in the scientific diagram there's a list of various structures that I want you to try to find and label on your paramecium. Feel free to use other resources to look at parameciums. There are many, many great videos and images on the internet that you can access easily, and they may all help you try to find some of these structures. Here's an artist image of a paramecium, and the structures that you're going to look for are the contractile vacuole, food vacuole, the oral groove, which is also sometimes called a gullet, and the macronucleus. You may notice there's a macro and a micronucleus on there. Paramecia actually have two kinds of nuclei. The large nucleus is called the macronucleus, and the small one is called the micronucleus. Both types of nuclei contain a full genome or a complement of genes. The macronucleus is the center of the metabolic activities, so it's transcribing proteins and enzymes that are involved in the paramecium's metabolism, and the organism cannot survive without the macronucleus. The micronucleus is kind of like a storage, or you can almost think of it as a gonadic part of the organism, where it's what gives rise to the macronucleus, and it's involved in conjugation, so exchange of genetic material with other parameciums, and it cannot reproduce without the micronucleus. Lastly, I want you to figure out what the motility structures are, and that's going to be part of your discussion for this report. Part B, the second activity, we are going to examine the swimming behavior of Euglena, a single-celled photosynthetic protist. The structures responsible for motility in Euglena are not the same as those used for the paramecium. Here are some pictures of Euglena that I took in the Biology 107 lab. They're a little bit smaller than the paramecium, and they're a lot faster. They're very hard to get good pictures of. These were some of the best ones that I was able to get, and unfortunately, I was wishing they were a lot better. You can see on the left, they're green, they have chloroplasts. The one on the right is I stained it with iodine, and you can see lots of structures on the inside, and it looks a little bit longer, and you'll see in the videos that Euglena, when they move, they kind of stretch and squish, stretch and squish. So sometimes they look a little longer and skinnier, sometimes they look a little bit fatter. There are 3 videos I have of Euglena. Here's one of them, and like I said, you'll be able to watch the swimming behavior of them. Some of the videos show a much closer view of the Euglena, and I'm hoping what you can see is the motility structures on these. Maybe you can just make it out in that video. Like I said, one of the videos has a really nice close-up, and you can definitely see the motility structures. So like with the paramecium, what you're going to do is also measure the size and draw a scientific diagram of Euglena. So there's a diagram there. They're a little bit too small to show the entire depth of the field, so you have to use exclusively the ocular micrometer to measure the size of the paramecium and enter that in table 5-1. Here's an artist drawing of Euglena. I'll just point out some of the structures that you're going to be looking at in the Euglena. So the nucleus, hopefully that one is obvious. It is usually the largest organelle, although not pigmented and colored. Also the chloroplasts. The chloroplasts, of course, are green. And something I'd like to point out is the eye spot. So the eye spot, you can see, is labeled right here. And it is pretty visible on a lot of the Euglena cells. It's kind of a reddish, brownish, orangish color. The eye spot apparatus is basically a photoreceptive organelle. So this allows the Euglena cells to sense the light direction and intensity and respond to it. They want to swim either towards the light or away from it. And these are photosynthetic, so probably they're trying to swim towards light unless it's too bright, would be my guess. You can see it has a number of other structures on there. They may not be visible on the Euglena cells. They're very small. Things like the contractile vacuole or the Golgi or whatever. And lastly, of course, you're going to identify the motility structure, which I think is pretty obvious on this particular diagram. In the third activity, we will examine the motility of Pelomyxa carolinensis. This is a large amoeba that can actually be seen with the naked eye. If you take a look at this diagram, this is actually viewed under the 10 times objective. This thing is huge. I'll show you another picture in a moment that shows exactly how huge this is. Here's another picture I took. This is under the 40 times objective. This was so big I could not fit the entire amoeba under the same field of view. If you take a look at this image, you can see it's actually 2 images that have been spliced together in order to fit the entire amoeba in there. So these things are massive. One structure I'd like to point out are the pseudopodia, or pseudopodium if you're just talking about one. So pseudopodia are temporary extensions of the cytoplasm. They're used in locomotion and feeding. So in locomotion, they're basically extending their cytoplasm and somewhat crawling along the surface or crawling through the medium in which they're living. During feeding, the pseudopodia can actually flow around and engulf prey and trap it in a sticky mesh and bring the prey in by phagocytosis. There are some great videos of amoeba on the internet, tons and tons of them showing phagocytosis. In this exercise, though, I want you to look at videos of amoeba basically moving around. This is not a video, this is actually some more photos that I took of amoeba under the 10 times objective. You can see here I took several photos. This was about maybe 10 or 15 seconds apart. You can see those pseudopodia changing shape as the amoeba is crawling along the slide. I thought these were some really cool pictures that I took. Here's a video I took of an amoeba, and you can see the cytoplasm is basically flowing. You can see that pseudopodia on the right is extending as the amoeba is trying to crawl across the glass slide. So two of the videos there are videos that I took. They're very nice. If you look carefully, you might be able to make out the nucleus. The nucleus kind of looks like a large transparent organelle in there. The pseudopodia, of course, are pretty obvious since they're all over the place. The third video is something that was done that was a little bit more professional with really expensive looking photography equipment, and is a very, very beautiful video. And you can see the amoeba moving that and maybe make out a few more details. So for the amoeba, I want you to measure the size, and I want you to draw a scientific diagram. Here's the picture of an amoeba that I mentioned. I would show you just how big these things are. If you take a look in the top right corner, there is a paramecium. So you get an idea how big an amoeba is compared to a paramecium. In fact, amoebas will eat the paramecium for food. And I believe that is actually why they're in the same jar with the amoeba, is so that the amoeba can actually have some nutrients. It's really unfortunate you are not able to be with me in the lab and to take a look at these. These specimens are very fun to look at. This lab is pretty low stress and usually is a lot shorter and quicker than all the other ones. Mostly you're observing behavior, answering some questions, measuring size, and drawing some diagrams. So check out the amoebas. Like I said, there are lots of really cool videos on the internet if you're really interested in these things. They're all showing different eukaryotic organelles and cell features, although, like I said, today we're focusing on motility structures. So here's a diagram of an amoeba, and like I said, there's some structures that you can make out. You can probably make out the nucleus in some of the images or videos. Definitely you can make out pseudopodia and maybe sometimes food vacuoles. A lot of those small things that you're seeing are actually small food vacuoles all over the cytoplasm. I thought I would share a little cartoon with you just for some humor for today. You can see it's from Dr. Fun, and it says in the caption, "Recent excitement in the microbiology lab ended abruptly when Dr. Rusco's giant amoeba turned out to be a fried egg." Ha ha ha. Thought you'd enjoy it. Now this brings us to the end of Lab 5. You may still have some YouTube videos to finish watching and some diagrams and other exercises to do, but these are the questions that you are responsible for Lab 4 and 5 in the Lab 4 or 5 short report. This is due 1 week after completion of Lab 5. If you have any questions, please get a hold of me and I'll see what I can do to answer your questions. Other than that, I hope you enjoy the videos and I will see you in class.